Hi guys. Okay, so in this video, it is the second part of chapter 1. We're going to be talking about the fifth of the 5 characteristics of life, which is called evolution. And we're also going to talk about the scientific method. And this should be a short video, so just hang in with me. We're going to watch a few videos that'll make it a little bit longer, but they're really good. I really like them. If you're one of my students from BISC 102, you have already seen these. Okay, so I'm going to share my screen and we are going to go ahead and finish this up. All right, so the 5th characteristic of life, we had already talked about organization, energy use, maintenance of internal constancy, which was homeostasis, reproduction, growth, and development, those 2 different types of reproduction. Now we're going to talk about evolution. Okay, just a genetic change over time is all evolution is, which we will talk about right now. So in this picture, you can see that this snake blends in incredibly well with its environment. Okay, so snakes in general that can blend in the best are going to catch the most prey because they are basically hidden in their environments. Because they're hidden in their environments, they're also going to survive the longest. And because they're surviving longer and eating more, they're going to reproduce the most. Okay, they're going to be the most physically healthy and physically able to reproduce again and again and again. That would mean that if you are well suited for your environment, I want you to write this down. The better suited for your environment you are, the more prey you get, so the more you eat, the healthier you are, the more likely you are to survive. And if you survive longer, you're going to reproduce more. Okay, so eat more, survive longer, reproduce more. If you reproduce more, your genes are going to be present more often in the next generation than in the same environment if you had an orange colored snake. Okay, an orange colored snake, what would happen to it? Would it live very long? Why? Well, unless it was a thing to its predators, okay? So unless like a hawk was coming to eat it and like got sick from eating it because the snake had these toxins or something like that, it wouldn't live the longest out of these 2 different colors, right? Out of the sand color and the orange color, just because it didn't blend well into its environment. So can an individual evolve? No. An individual, you are an individual and I am an individual. Can we evolve? No. Can we like grow and develop? Yes. Can we like change our attitudes and opinions and circumstances and like living arrangements? Yes. All of those things can happen. Can we move? Can we migrate? Can we like do all of these beautiful, amazing things? Yes. Are they evolution? No. Evolution is a genetic change over time, over many generations. So can an individual evolve? No. That would be like me saying, I get dropped off in the ocean and left. The boat just leaves. Am I going to be able to say, wow, E, it would be really, really freaking useful in order for you to grow gills right now? That would be incredibly useful. Could I do it? No, because an individual cannot evolve just because it would be like useful, just because that adaptation would be advantageous. So what is the source of variability in a population? If I had this sand-colored snake and an orange snake, what is the source of that variability? It's its genetics, okay? Your genes. So the difference between me and you, our hair colors, our heights, our eye colors, our skin color, anything between us are our genetics, okay? Hold on. All right, so if nothing ever changed in the environment you are in, so I'm in Oxford, Mississippi. In Oxford, Mississippi, let's talk about sickle cell anemia, this one first. So there is an advantage to having sickle cell anemia if you live in a country that is because sickle cell anemia, if you have sickle cell anemia, sucks because you have anemia, okay? It's like not great, but you're not going to die of malaria, okay? gave us sickle cell anemia be advantageous if nothing ever changed in the environment that you are in right now? If you're not in Oxford, Mississippi, you're in Jackson, you're in Grenada, you're in Colorado, you're wherever the hell you are, okay? In your community, are you being ravaged by malaria? No, probably not. So nothing ever changed. Malaria is not affecting you. Would it be advantageous to have sickle cell anemia? No, because you're already not going to likely get malaria. So it doesn't matter that you would be resistant to malaria. It would just suck that you have sickle cell anemia, which a lot of our students at Ole Miss, plenty. Every single semester I have students coming up to me telling me that they have sickle cell anemia. So no, that gene would not be advantageous if everywhere in the world you would be, it's dependent on the environment that you're in. Just like that snake. If that snake wasn't living in sand, okay, if it was living in, if it had orange sand, if it was a completely different colored sand, all of a sudden that orange snake would be better suited for that environment. Okay, I hope that makes sense. Another one, evolved as rapidly if humans had not consumed antibiotics. If there were no antibiotics, would antibiotic resistant bacteria have evolved? I mean, that trait would have evolved. Okay, that trait would probably be around in a certain subset of the population. But if there were no antibiotics, would it be relevant to the bacteria? No, because they would be living large anyway, whether or not they had antibiotic resistance or not, because there were no antibiotics. Okay, so again, it's about the selection pressures in a particular environment. If you have an environment that doesn't have malaria or does not have antibiotics, then sickle cell anemia or antibiotic resistant bacteria, they don't pose the same sorts of advantages as in other environments. Okay, so natural selection is based on specific selective pressures put on a population. Specific selective pressures put on a population. All right, this video is very dull. We are going to, I hope y'all can hear this.
At a glance, it's apparent that no two individuals are resistant to antibiotics, resistance and bacteria. How occurs in one species of bacteria, stand the origin to prevent from developing. Let's explore how a population of bacteria might become increasingly resistant to antibiotics. Suppose that all individuals in a population are vulnerable to antibiotics, as they are in generation 1. Over time, mutations will occur. Some of these mutations might confer antibiotic resistance, resulting in variation in antibiotic resistance among individuals in the population. When the antibiotic is administered, most of the vulnerable green cells are killed, whereas the more resistant red cells survive. The population has now shifted to a higher proportion of antibiotic-resistant individuals. When this new population reproduces, more individuals will be antibiotic-resistant than the previous generation. It's important to realize that natural selection works on the variation already present in a population. For example, if all individuals in generation 2 were equally vulnerable to antibiotics, then administering antibiotics would not change the frequency of resistance in the next generation. That is, antibiotics do not create antibiotic resistance in the bacteria. Rather, a random mutation occurred before the bacteria were ever exposed to antibiotics. Natural selection favored the resistance allele once antibiotics were present. Antibiotic-resistant bacteria only have an advantage in an environment where antibiotics are present. If antibiotics were not administered after generation 2, then the frequency of antibiotic resistance would not have changed in the bacterial population. In other words, the traits favored by natural selection depend on which selective forces are acting on the population. Although the origin of variation in a population is mostly random, natural selection is not random. Within each environment, individuals with adaptations that increase survival and reproductive success have a selective advantage. Understanding the basic basics of natural selection, explains why species seem to have perfect traits for their environment, such as antibiotic resistance in bacteria that are often exposed to antibiotics.
Okay, so guys, if we're looking at that, what she said is that this mutation had come about before antibiotics were administered. Okay, this population already had this mutation in it that conferred antibiotic resistance. That meant if antibiotics came around, this particular bacterium would be resistant to that. It would not die if it was exposed to antibiotics. Okay, so if antibiotics are present, that means that this bacterium is going to thrive. Almost everybody else is dying, and it is thriving. It is going to reproduce so much that it is present. This trait, this red trait, is present more often in the next generation than the green trait. That's because it conferred this sort of selective advantage in that particular environment. Now, would it always be the case that this resistant bacteria was favored? No. If antibiotics were not present, if they're absent, there is no reason that this would survive any better than any of the other ones, because the other ones would not die from antibiotics. Okay, so selective pressures are what's important here. Specifically with antibiotic resistance, evolution can act more quickly when there are many generations. So if you look at some bacteria, it is super easy to action with bacteria because they reproduce asexually. Very little genetic variability, and the variability that is there in an asexually reproducing species is random. It's just from random mutations. So the more generations that you have, the more that they reproduce, because they don't need to find a mate, they don't need to copulate, they don't need to make sex cells, they don't need to do anything. They just need to like bud off and make a new one. Okay, so the more generations you have, that means that you have an increased probability of these genetic mutations, these random mutations that will affect the fitness of individuals in the population. All of a sudden you have a random mutation. Most of them will not do anything. Most of them will not be beneficial or like negative, affect them negatively, but sometimes they do. Sometimes you get a random mutation and it's antibiotic resistance. Well, that's great because it's going to live a long, lush life. So antibiotic resistance or evolution can be observed much more quickly in something like bacteria than it can in complex organisms like humans, which we can observe only over thousands and millions of years. Okay. All right, we are going to watch this video, and I have to switch over the screen that I'm sharing. Okay. I hope you all can see this.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, porous and thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
All right, guys, so hold on. Oh, well, all right, goodbye. So let me stop sharing this with you. Okay, so 11 days. 11 days to go from no antibiotic to 1,000 times. Oh shit, hold on. Hold on. Okay, sorry. 1,000 times the amount of antibiotic that a typical bacterium can withstand. Okay, that's insane. That is absolutely terrifying. And if you are not scared, you're silly. Okay, and you're lying to yourself and you don't understand it. Can an individual evolve? No. So those individual bacteria, as they're going, he said they have to develop a new mutation to get them to the next column. Can a bacterium, just develop a new mutation? No. But in that like community of bacteria, if one has the ability to move on, it will. Okay, it will. Instead of being just crowded in that little area, if it can move on to the next area that is less crowded, it can go ahead and reproduce as much as it wants. It's going to move on. It's going to move over there. Okay, so it does. So they acquire those successive mutations. That is crazy. Okay, here is a video about finches. I'm going to let y'all watch this.
Peter and Rosemary Grant have been studying the Galapagos finches.
Those shorts,
they measured and banded every single finch on the island of Daphne Major. In 1977, the island experienced a terrible drought, receiving less than 20% of the rainfall in a normal year. The finches preferred a diet of small, tender seeds, but these seeds became harder and harder to find. The finches were left with large, hard seeds. Because they couldn't eat the large seeds, they began to die in large numbers. In a single year, 85% of the finches died. The Grants discovered that the surviving birds had larger beaks and were able to eat the larger seeds. The survivors then passed this trait on to the next generation. Here was real evidence of evolution by natural selection.
Okay, so evolution by natural selection. What was the selection? What was that selective pressure? It was the drought. If nothing had ever changed, would all of those finches have died? No. If that drought had never happened, would the finches had to have had these larger, harder beaks? No, but based on the environment that they're in, based on what is changing in that environment, sometimes, you know, the individuals that are best suited for that environment are going to be the ones who live. And if they live, because they're able to eat these larger, harder seeds, they're going to pass that trait onto their offspring, and their offspring will also be able to live. So the traits that are favored in an environment, like that sand-colored snake, are going to be more present in the next generation. Okay, and we're going to continue talking about this through the entire term. So 4 weeks. Evolution is literally all it is. Guys, it is 2020. Holy cow. There's no, I don't believe in evolution anymore. This is not a thing to believe or disbelieve. It is a fact of nature. It is literally a fact of nature. All it is a change in genetic makeup of a population over many generations. Think about it. If you have all of these finches dying and only a few left, those few left are going to reproduce, and their alleles, their genes, are going to be more present in the next generation. That is a shift in the genetic makeup of a population over a generation. That's evolution in action. All it is this change in genes over time that could lead to these changes in traits over time, okay? But it's all about the genes. It's all about just that change in genes and populations over time. Can an individual evolve? No. All right, we're going to talk about the scientific method. I'm not going to make this painful because you've heard about the scientific method probably 300 times. So, guys, in general, all scientific inquiry is going to follow a standard process. You're going to make observations, ask questions, consulting that prior knowledge. You're going to formulate a hypothesis, make predictions, collect and interpret data, and then consult that prior data to make sure that your data is making sense. Draw conclusions. You're going to peer review manuscripts, and then you're going to publish that manuscript. Okay, so meaning that your data will now be available to others and should be able to be replicated. So scientists test their hypotheses with discovery science, meaning that they are viewing things as they're taking place. So for instance, this woman, if she's studying migratory birds, that's not something that you can really do in just a controlled laboratory setting. Okay, so that's discovery science versus controlled experiments. So if they're doing, it looks like they're doing something with coffee. But say that they were doing a controlled experiment with plants. Okay, that's a controlled experiment where they are able to manipulate or control different variables. Versus discovery science, you're literally just able to report what you see. You're not able to, and you don't really want to change an environment. Happens. All right, so you observe some tomatoes are larger than other tomatoes, and you ask a question. All right, some are larger than others, so does sunlight affect the size of these tomatoes? You hypothesize that the more sunlight a tomato plant receives, the larger the tomatoes will grow. That is reasonable. What have you based this hypothesis on? You've based it on what you've observed, which is that something is causing some tomatoes to be larger than others, and you're also basing it on your prior knowledge. What is prior knowledge that you or anyone has with plants? That they need nutrients, they need sunlight, they need soil, they need water in order to live, okay? So you're basing it on prior knowledge. Your prediction is that increasing the amount of sunlight in my experiment will, like in one plot of plants, will cause an increase in the size of those tomatoes compared to this other crop of plants that receives less of that sunlight, okay? So if you have like one of your groups is shaded, one of them is in full sun, you would expect that the full sun plants would have these larger tomatoes. So you would, whoa. Sometimes my brain is going ahead of my mouth. Okay, so you design a fair experiment that will control for variables to make the experiment as consistent as possible. And we're going to talk about your variables. Your variables are super, super, super important. If you are doing a controlled experiment, you need to control as many variables as you possibly can, okay? So you're gonna have independent, dependent, and standardized variables. Your independent variable is what is manipulated. So if one group is receiving a ton of sunlight and one group is receiving less sunlight, you are manipulating the amount of sunlight. If one group was receiving more water, one group was receiving less water, you're manipulating the amount of water. If one was put in a 4-gallon pot and one was put in a 4-inch pot, you're manipulating the amount of soil, okay, or the size of the container. So then your dependent variable is whatever is measured. Now, you can have many dependent variables. You can measure, you can say, okay, well, I'm getting larger tomatoes because of this increased sunlight. Am I also getting more tomatoes overall with this increased amount of sunlight? So you can have multiple things that you're measuring, like the size or the number or the height of the tomato plants, but should you have more than one independent variable per experiment? No, okay? If you had different groups in an experiment, fine. But in a group, you need to hold constant as many things as possible. And that's where your standardized variables come in. So the only thing that should be changed in between all of your different experimental groups is your independent variable. That's it. That's all that should ever change. So you need to hold these things constant for all subjects in an experiment, meaning you need to have the same amount of water. They need to be watered at the same time of the day. They need to be watered like, well, okay, on the same schedule. They need to be in the same size pots. They need to be the same variety of tomato plants. They need to, all of these things, okay, need to be kept as constant as possible. That way, at the end of your experiment, if you see differences in between your experimental and your control groups, guess what? You can say, because you controlled for everything, you can say, okay, the thing that changed the size of my tomato plants was definitely the amount of sunlight, okay? So holding your standardized variables the same. Now, standardized variables are different than your control group. Your control group is where you like don't change the independent variable, okay? Or it receives like nothing, or like your control group, it's typically like, so for instance, if it was amount of sunlight, you wouldn't put it in no sunlight, but like you could put it in no sunlight, I guess. It just wouldn't grow, but like a shaded plant, okay? Or whatever, okay? Your control group is just another group where you're measuring something, right? But your standardized variables, they're going to be the same regardless of whether you're in the control group or whether you're in the experimental group, okay? Okay, so then you're going to collect and interpret your data, recording all data accurately, going to draw a conclusion. You can reject the hypothesis, or you cannot reject or accept the hypothesis. So rejecting or accepting the hypothesis. Now, what makes a flawed experiment? If you have multiple independent variables. So say you manipulated the amount of water and the amount of sunlight in the same experimental groups. Well, and then you found that there was a difference between the size of the tomato plants. Would you know if it was due to the difference in the water or the difference in the sunlight? No, because they're like confounding with each other. So flawed experiment, you're trying to make a shitty experiment, you're going to have a ton of independent variables that you have manipulated in your experiment. You would not have many standardized variables. So for instance, if you had you're just using what you had, and you're putting one of your plants in a 4-gallon pot, one of your plants in a 1-gallon pot. You're watering at different times of the day. You're not measuring how much you're watering. All of those things. Not gathering your data consistently. All of those things, or gathered by different people. Like all of those things would make a flawed experiment. All right, guys. Stop share. Guys, that's the end of chapter 1. Congratulations. So either today or tomorrow, I'm going to record for chapter 12. Okay, so chapter 12, we'll be talking a whole lot more about evolution. So I was gonna say get your shoes on, like, I don't know, get ready.